case review. For tonight, our webinar will include 9 research presentations on wound healing, and the session will be moderated by Dr. Henry Shaw. Before we begin, I would also like to highlight that following completion of tonight's program, we will also be holding an informal 30-minute question-answer session or so for all our residents, fellows, and medical students, attendees that have logged on tonight, for them to get some informal career or research advice. So I would like to welcome all active members of the Research Council, including members of the Mentorship Committee and all their mid and senior career members who are on tonight, to stay on for a little bit, and we can hopefully set up some smaller breakout rooms if we have enough people to have an informal mentor session. Finally, I would like to thank 3M for generously providing support to sponsor this 4-part series, as well as the interactive symposium. Joseph Gugliuzzo, I think, is on from 3M tonight. So thank you for generously providing support to really sponsor this 4-part series, as well as the interactive case symposium. So now we'll move on to the research presentations for the evening. There are 9 5-minute talks that we've lined up tonight. We'll have the first 5 presentations, followed by 10 minutes of discussion. And then the final 4 talks followed by a little more time for discussion. Please hold your questions until the designated time for discussion, or you can also enter them into the chat box and I'll keep an eye on them. So without further ado, I'd like to hand the session over to our moderator tonight, Dr. Henry Shaw.
Thanks, Derek. And as Derek said, I am Henry Shaw, and I'm very happy to be here moderating, and I appreciate the invitation, and I'm very excited to introduce these 9 fantastic papers here. And so without much further ado myself, we'll get started. And I apologize ahead of time if I mispronounce any names here. So our first paper is entitled Transdermal Deferoxamine Alleviates Radiation-Induced Damage in Porcine Skin, and will be presented by JUN-DEPENDENT from Stanford University.
Great. Thanks for the introduction. So, hi everyone, my name is JUN-DEPENDENT, and I'm oh, whoops.
Yeah,
I have to share my screen. Whoops.
And people should just go ahead
and
introducing you. Yeah, to share your, feel free.
Gotcha.
You don't need an invitation to share.
Let me just do that real quick. Guys can hear me?
Yep.
Screen good? One sec, let me, looks good?
Yeah, yep, you're good to go.
Great. Oh, whoops, it's malfunctioning. Sorry guys, technical difficulties. All right, cool, all right, all good. So, hi everyone, my name is JUN-DEPENDENT, and I'm a research associate who works under Dr. Wan and Hagee Laboratory at Stanford. And in this presentation, I'll be talking about a novel transdermal drug delivery system containing DEFEROXAMINE and how it reduces radiation-induced damage in pig skin. I have no disclosures to state in this presentation. Radiotherapy is a staple treatment modality for cancer, but one of its largest drawbacks is that it causes significant damage to surrounding soft tissue, which may further advance into radiation-induced skin fibrosis, leading to hypovascularity and dermal induration. DEFEROXAMINE, or DFO, is an FDA-approved iron chelation drug, and it is previously shown to improve skin vascularization when injected into radiated tissue. When DFO chelates iron, it stabilizes hypoxia-inducible factor 1 alpha, or HIF1 alpha, enabling it to translocate into the nucleus and act as a transcription factor for a number of proangiogenic genes, which results in improved tissue vascularization and regeneration. Local chelation of iron also reduces levels of reactive oxygen species, which are known to play a role in the pathogenesis of radiation fibrosis. However, repeated injections of DFO to radiated skin can damage the soft tissue and cause patient discomfort. To solve this problem, we've developed a transdermal drug delivery system to provide cutaneous delivery of DFO without the need of an injection. So I'll go over the mechanism briefly. So DFO is first encapsulated in reverse micelles within a biodegradable patch. When applying the patch topically to the skin, the reverse micelles first penetrate through the hydrophobic outer layers of the epidermis and then eventually disintegrate into the more hydrophilic environment of the dermis, where the DFO is then released. To test our DFO patches, we irradiated 3 red Dirac pigs with a single fractionated dose of 30 gray. and to localize regions on each side of the pigs. Pigs were allowed to recover for 8 weeks, allowing radiation damage to develop, and they were then divided into 3 treatment conditions: a pig treated with the DFO patch, a pig with the control patch with no medication, and a pig with no patch at all to serve as control. Weekly biopsies and cutometer readings were taken from each group with their respective adjacent non-radiated skin. Our final cutometer results show that DFO-treated skin had the greatest deformation in comparison to control and no patch conditions, indicating that DFO-treated skin was less stiff and showed biomechanical improvement. Histological sections of skin biopsies were stained with hematoxylin eosin to evaluate the dermal thickness, and DFO-treated skin demonstrated significant reduction in dermal thickness compared to both control and no patch groups. Skin sections were also stained with picarocetus red to evaluate collagen deposition. DFO-treated skin had significantly lower collagen content compared to no patch and control patch-treated skin. The vascularity of skin was determined by CD31 immunofluorescence staining, and here the DFO treated skin demonstrated significantly higher vascularity compared to control and no patch treated skin. So in conclusion, transdermal DFO delivery offers an effective and non-invasive mechanism to improve perfusion and reduce stiffness of irradiated pig skin. And as pig skin closely resembles human skin, our findings establish incredible potential to treat patients with radiation-induced soft tissue defects. And I would like to thank everybody at Hague Lab and Stanford VSC for making all this possible. Thank you.
Great, thank you. And thank you so much for being on time too. I really appreciate that. And our next speaker can go ahead and start to share while I do the introduction. Our second paper for this evening is entitled CD36 Antagonism Minimizes Skin Scarring by Inhibiting JUN-Dependent Fibrotic Pathways within Fibrogenic Fibroblast Subpopulations. And it will be presented by Mimi Borelli from Stanford. Are you okay there, Mimi?
Hello, yes, sorry, just trying to make it work.
Okay, no worries.
Okay. Can you see this?
Not yet.
Okay, sorry.
Yeah, it's okay.
Can you see this?
Can you see this?
Not yet, no. Are you sharing, are you picking the window? The actual.
Oh, okay, yes, I think I've got it now. Can you see this?
I see your desktop, your entire.
Okay. Slideshow, here we go. How's this?
It right now is just showing your desktop with the maybe there's a delay or something, but it so if you unshare and then right now it's just showing your desktop.
I have nothing to disclose.
Okay, hang on.
Skin fibrosis is the end result of...
Sorry, new share.
Yeah, we do a new share and but you have to so
does
at
this
least
work?
We developed a trend in this work.
Sorry, 1 second. I think the problem is I recorded it to practice, and now I don't know how to do the version that I didn't record. Does anyone know how to do that on a Mac?
Henry, why don't we move to the third talk, and
Okay.
then Mimi, why don't you email April and the rest of the PSRC staff your PowerPoint talk, and then they can present it and share for you.
Okay.
Okay, sounds good. All right, so Kellen Chen will be our next presenter. All right, so Kellen, why don't you go ahead and share your screen while I introduce you. So Kellen's paper is going to be disruption of mechanotransduction signaling preserves fibroblast heterogeneity and promotes tissue regeneration in healing wounds. And Kellen also is joining us from Stanford.
Thank you very much. I'd like to thank all of my lab mates, especially Dr. Gertner, with all their help with conducting this research. So a little bit of background. The unmet clinical need we are trying to address is that injury to the skin from trauma, burns, radiation, and surgery can lead to hypertrophic scar formation. So on the left here, we see a 17-year-old male that had a flash burn and 6 months later had a lot of hypertrophic scar formation all over his face. Currently, there's no effective therapies for these deep partial thickness injuries, especially those over sort of uneven contours or large surface areas. Over the past decade or more, our lab and other labs have also found that focal adhesion kinase inhibitor, or FACI, can be used to basically modulate the mechanotransduction of cells and basically improve scar formation, mostly in small animal models. So here we sought to look at the ability to help fibrosis in a large animal model. To do this, we created 8 wounds on the pig dorsum, and we had 3 treatment groups. One was our normal wound, which would create scar. The next is wound with a hydrogel that just on its own. And finally, we did a wound that had a hydrogel treatment that had FACI also within it. And the idea behind this is that it would be able to keep a constant delivery of the drug and also provide additional therapy. So here we have a schematic just showing our FACI sort of imprinted in the hydrogel, and it diffuses down into the injury site over time. So due to it being a 5-minute talk, we were going to talk about the results at a little more of a high-level view. But overall, what we found was that disruption of mechanotransduction accelerates wound healing, attenuates fibrotic scar formation, and promotes tissue regeneration. So here we again see our 3 groups. We have our wounded group that creates scar, our wound with our sort of blank hydrogel, and our wound with the hydrogel with FACI. We see that over the course of 180 days, we have improved scar appearance and also the potential regeneration of some hair follicles. Here we quantify wound closure rates. So the hydrogel and the scar groups are in gray and blue respectively, whereas the FACI group is in red. We see that the FACI group closes their wounds much faster, and here we've quantified this, showing that the wounds close roughly at 15 days compared to 25 days in the other 2 groups. We also used a VAS score, which is a scar appearance score, to show that the FACI treated group also had improved scar appearance. A lot of these results have been shown previously at other iterations of this meeting, and there we've also shown with other trichrome staining and picroserous red staining that there is indeed more fibrotic formation of collagen in the wounded group that is attenuated, and also that there are some hair follicle regeneration with the FACI treated group. For the purposes of this talk though, I'm going to move on to the next part of the study, where we try to actually find out what this might actually be doing to our cells. So to do this, we've tried a variety of different methods, and in this talk, I will talk about our in vitro study. Here, we isolated human dermal fibroblasts to explore the clinical relevance of this therapy and sort of what it might do to actual human cells. So we have a 3D culture system where we basically can imitate the scar environment, and we have 3 groups in which we have our control group, our strained group, which sort of activates the cells like a scar environment, and then our strain plus FACI, which is sort of our treatment group. Then from these 3 different patients, we did our study and submitted them for single cell sequencing to look at the genes that the cells were displaying. Here we can see that we have our 3 groups, so strain and plus FACI, the no strain, which is control, and strain, which is like our scar group. And we're able to show all of our different cells in this UMAP embedding. And on the top, I show the 3 individual patients in their sort of individual spaces. And what you can see here is preserved across all 3 patients, we have this behavior where strain activates cells into this sort of new genetic phenotype, whereas the treatment with FACI shifts them back over to the left here. And if we look specifically at fibroblast heterogeneity, which has become a more popular topic in the literature, we see that our control cells cluster mostly in one cluster here in cluster 0. Strain activates a lot of fibroblast heterogeneity in these 5 clusters here, 2, 4, 5, 7, and 8. And finally, treatment with FACI reduces some of this heterogeneity and shifts them to a sort of new metastate over here on the left. We interrogated a variety of different genes and pathways. For this talk, I'm going to just talk about some of the pathways we used. Here we're using GeneTrail to sort of visualize our pathways. We can see that strain activates commonly accepted mechanotransduction markers such as focal adhesion, MAPK, EGFR, and also immune response activation, whereas the FACI treatment seems to cause cellular detoxification, ECM disassembly, activation of MMPs, which are enzymes that break apart collagen and scar tissue, and also epithelial cell migration. So overall showing a more regenerative phenotype that's less of this activated scar collagen depositing phenotype. So overall, in sort of this 2-part study of in vivo and vitro, we showed that disruption of mechanotransduction accelerates wound healing, attenuates fibrotic scar formation, and promotes tissue regeneration. We attempted to look at human relevance in fibroblasts and demonstrated that the treatment actually does shift the cells on a gene level toward more pro-healing and anti-fibrotic phenotypes. And overall, these 2 studies combined give us a lot of confidence potentially to use this therapy to prevent HTS in humans in the future. Again, I'd like to thank everyone in my lab and my funding sources, and thank you very much.
Great, thank you very much, Kellen. Our next speaker will be Abra Shen. And Abra, go ahead and start sharing your screen. And her talk will be entitled "Deferoxamine Treatment Decreases Levels of Reactive Oxygen Species and Cellular Apoptosis in Irradiated Skin." And Abra is also speaking from Stanford.
Hi, thank you, and good evening. I'm very grateful for the opportunity to present our work. My name is Abra Shen, and I'm a 4th-year medical student at Harvard. I had the great opportunity to work with Dr. Wan and Dr. Longacre at Stanford on this project. Transdermal deferoxamine treatment mitigates fibrosis and irradiated skin. Millions of patients are diagnosed with cancer each year, and many will eventually receive radiation therapy. Unfortunately, radiation can lead to soft tissue damage, which can lead to functional and aesthetic deficits. Radiation therapy is associated with acute rises in reactive oxygen species, or ROS, and apoptotic proteins, and this damage may progress to radiation-induced fibrosis. Radiation-induced fibrosis is associated with hypoperfusion and tissue hypoxia. Interestingly, iron plays a critical role in catalyzing the formation of reactive oxygen species. And so we examined deferoxamine, or DFO, an FDA-approved iron chelator that reduces levels of iron and tissue hypoxia. The goal of the study was to investigate the impact of deferoxamine treatment on radiated skin in a mouse model. We hypothesized that because iron catalyzes the formation of reactive oxygen species, treatment with the iron chelator deferoxamine would reduce levels of ROS, improve tissue vascularity, and mitigate radiation-induced fibrosis. I will quickly go over the mechanism of DFO, as my colleague described it very well earlier today. DFO works by decreasing iron levels, which increases levels of HIF-1 alpha, and then HIF-1 alpha is able to target genes such as VEGF to promote angiogenesis. In this study, we evaluated deferoxamine delivered as a transdermal patch to serve as a potential therapeutic for radiation-induced skin fibrosis. This is what the patch looked like. CD1 nude mice were randomized to one of 4 groups: irradiation only with no treatment, DFO treatment post-irradiation, prophylaxis treatment with DFO treatment starting 2 weeks before irradiation and continuing past it, or finally a control group which received no irradiation at all. The transdermal patches were placed on the mice's scalps and replaced daily. We first examined levels of reactive oxygen species and apoptotic proteins. Immediately after radiation, mice were sacrificed and were stained with dihydroethidium, an ROS indicator, as shown on the left, and ELISAs were performed to measure the apoptotic markers BAX and cleaved caspase 3, as shown on the right. prophylactic DFO treatment was associated with significantly decreased levels of reactive oxygen species and apoptotic markers. Since radiation therapy leads to hypoperfusion and hypoxia of the skin, we stain skin for CD31, a marker for endothelial cells. As expected, the skin of non-irradiated mice was significantly more vascularized than that of irradiated, untreated skin. And furthermore, the skin of mice in the DFO prophylaxis group was just as vascularized as the skin of non-irradiated mice and significantly more vascularized than the skin of irradiated, untreated mice. Finally, because radiation therapy leads to fibrotic and thickened skin, we looked at dermal thickness to evaluate whether DFO treatment could mitigate the fibrotic changes seen in skin post-irradiation. Dermal thickness was evaluated with hematoxylin and eosin staining as shown above, and collagen fibers were evaluated with picrocereus red staining as shown below. Both stains showed that skin treated with DFO prophylaxis was most similar to non-irradiated skin and significantly different from irradiated, untreated skin. In summary, prophylactic DFO treatment was associated with significantly decreased levels of reactive oxygen species and apoptotic markers, as well as vascularity, dermal thickness, and collagen fiber networks, which were most similar to those of non-irradiated, i.e. control mice. So in conclusion, transdermal delivery of DFO improves tissue perfusion and mitigates radiation-induced skin fibrosis, highlighting a potential role for DFO in the treatment of oncologic patients. These results are very promising and demonstrate that DFO prophylaxis may have a significant therapeutic impact for patients undergoing radiation therapy. Thank you very much for your time.
All right. Thank you very much, Abra. That was great. Is Brian Bennett on the line with us?
No, this is Suji Diaplina, so I will be presenting.
Oh, terrific. Well, yes, very happy to see you. Okay, so why don't you go ahead and share your screen and I will introduce you. So our next talk is entitled Evaluation of Heat-Treated Fluoridated Apatite Coatings for Improving Soft Tissue Wound Healing Around Percutaneous Devices, and will be presented by Dr. Jayapalina from University of Utah.
Thank you. I would like to take this opportunity to thank my co-authors as well as the Utah Percutaneous Osseointegrated Prosthetic Research and Clinical Team. As you all know, percutaneous devices connect the internal and external bodily environment, which allows therapeutic use of medicine as well as functional rehabilitation. Our interest is the rehabilitating the amputees with percutaneous osseointegrated implant. Traditionally, socket prosthetics are used for amputee rehabilitation. Oftentimes, amongst many shortfall, the amputee stump sides suffer from pressure sores and skin breakdown. Oftentimes, amputees have reduced prosthetic use, or some even abandon the socket system. Alternatively, percutaneous osseointegrated docking system allow direct transfer of ground reaction force to the skeletal system, which allows better gait and longer prosthetic use time. More importantly, it allows reduce donning and doffing time. You can see here, it only took 5 seconds for amputees to remove the prosthetics. Although it's a superior system, they are concerned. One is the superficial infection as well as the bone resorption. Especially this, you can see on the right-hand side, this system is a Swedish implant system. And you can see after 2 years of implantation, most of the unloaded bone or the bone below the screw is resorbed away, as well as the cortical wall thinned. What we have done is using the the bone remodeling principle and understanding the load, we made an end-bearing implant system, which allowed the forces to get transferred at the resected bone end, which prevented the distal bone resorption. However, skin implant interface is a different story. As the time proceeds, and this is one of our sheep model, you can clearly see the skin migrate down. The bottom panel shows the different time series. And if you, the top right-hand side, you can see the skin or epithelial downgrowth as well as the skin implant. The pocket is filled with debris. Eventually, it becomes needed for infection, and infection do occur. Regardless of the different animal model that we use, we see the interface as a wound where you see a granulation tissue and hyperplastic region, as well as the downgrowing thin epithelial. Unfortunately, one of our patients had infection approximately around 200 days after the implantation. Again, you can clearly see the hyperplastic and migrating epithelial edge as well as the granulation tissue. So, which shows us the what we see in the animal model is actually get translated in human. So we, a long time ago, we decided that it is a wound edge. You can clearly see the parallels here, right at the healthy dermal region. You see the hyperplastic region, and then you see the migrating epithelial tongue. However, this happened like 3 to 10 days after the wounding. In this particular animal tissue, we seeing the same phenomenon 9 months post surgical period, which tell us that our implant periprosthetic tissue is not healing completely or healed completely, and that's why we are seeing the downgrowth. So in order to find solution, we look at the natural interfaces, one of which is the enamel gingival epithelial interface. And if you look at the literature, you can see the junctional epithelial cells induces hemidesmosome. That hemidesmosome allows the cell to adhere to the enamel surfaces, allowing a tighter junction. So we wanted to produce that in our interface. So we made, so enamel is not the epitide, it's a fluoridated epitide. And different degree of fluoridation is found in different animals. So we made the flora epitide, which is both of the hydroxyl group is replaced with the fluoride, and then we replaced just only one. And when we actually did an in vitro study, we found that sintering actually helps. And the bottom panel shows the involucrin, which is the differentiation marker for epithelial cells, and especially the one which is sintered at 1150 produced the most differentiated keratinocyte that we seeded. So we undertook an in vivo study.
Can you wrap up in the next minute or so?
Sure. So we did an animal study, and we put the implant in the back of the animal, and we looked at the epithelial downgrowth and also the granulation tissue. And here you can see the uncoated implant that produces the most downgrowth, whereas the FA, you see hardly any downgrowth. And here's the interface. The white arrow shows the downgrowth, and yellow arrow shows where the epithelium was at time 0. And again, we did PCR it shows that different markers, especially the TGF alpha, is upregulated in FA-coated implant system, which actually prevents the migration of the transformation of epithelial cells into migratory phenotype. So in conclusion, yes, the FA actually facilitate the healing, possibly by introducing the hemidesmosome, which need to be further validated, as well as we have to do a weight-bearing animal model, which is currently funded by the VA. And also we need to understand the healing pathway. I just wanted to put the last slide showing the similarity between sintering and the natural tooth enamel SEM images. Thank you for listening.
Great. Thank you, Suji. It's very fascinating work. So we're going to go back to Mimi. Are you ready now, Mimi?
I'm ready.
Okay. All right. I will actually, I'll give you the title again. All right. And Mimi's talk today will be CD36 antagonism minimizes skin scarring. You can go ahead and share your screen while I'm reading this.
Great.
CD36 antagonism minimizes skin scarring by inhibiting JUN-dependent fibrotic pathways within fibrogenic fibroblast subpopulations. Right, take it away.
Can I confirm I'm sharing the right screen?
You are. I see it perfectly now.
Excellent. Okay. Good afternoon, everybody. My name's Mimi. I'm a PGY1 at Brown, and this is work that I did whilst spending time at the Longacre, Wernig, and Lorenz labs at Stanford. Okay. Now I can't move to the next slide. I have nothing to disclose. Skin fibrosis is the end result of injury in human skin. In the United States alone, over 100 million new scars are formed every year, and with no therapy currently able to prevent or reverse scarring, the medico-economic burden is enormous. We recently developed a transgenic mouse in which all cells have the capacity to express JUN under the influence of tetracycline, such as doxycycline. When we constitutively express JUN in this mouse, we find when we look at all organs under the microscope, there's evidence of fibrosis, including fibrosis of the skin. We want to investigate what exactly the role and mechanism by which JUN expression leads to scarring in the skin. To do this, we first derived primary cultures of human fibroblasts from normal unwounded skin and from scarred hypertrophic scarred skin. We used a CRISPR-Cas9 method to delete expression of JUN in these fibroblasts and then compared the downstream effects on their epigenetic landscape and gene expression. When we did this, we found that knocking out JUN had a profound effect, mainly on genes that were highly involved in fibroproliferative pathways, as shown here, ECM receptor interaction, PPR gamma signaling, and focal adhesion. We focused specifically on the genes that were both down-regulated and had closing landscapes, of which there were 106, and interrogated this list to find a good gene that would be available as a surface marker for us to interrogate, linked to JUN and fibrosis, and was able to be antagonized. And when we did this investigation, 36 came up as the best candidate. 36 is a glycoprotein transmembrane receptor. It's also known to be a fatty acid transporter, and it's been shown in previous work to be associated with fibrosis, but not with JUN. We showed in our fibroblasts, so the FSP1 expressing cells that are fibroblasts, that we did find expression of both JUN and CD36 at the protein level, and there was close association between JUN and 36. As you can see on the right, the 36 expression is perinuclear as well as on the surface. It's quite faint in this picture, but it is there. And JUN is in the nucleus as expected. Finally, and importantly, there was an available water-soluble molecule called salvinoleic acid, which antagonizes CD36. It's actually from the red root of the salvie plant, shown on the right. So we wanted to look at whether we could use salvinoleic acid to antagonize CD36 and therefore interrogate and disrupt the JUN pathways and decrease scarring. We first of all did this in our cells in vitro, and as you can see here, delivery of salvinoleic acid to the cells decreased fibroblast proliferation, collagen production, and TGF-beta production. Next, we wanted to see what the effects would be of delivering salvinoleic acid to mice in an in vivo model. We used our JUN mice and used doxycycline to, at this wound base so that the wounds would turn into hypertrophic scars. And then we treated a subgroup of these mice with salvinoleic acid and looked at the outcomes on the wounds that they formed. As you can see in this picture, the mice that were treated with JUN had a reduced size of their wound. It was less raised and it was less red. The graph on the right shows that the overall wound healing times did not change between salvinoleic acid treatment and no treatment. However, when we looked at the wounds histologically, salvinoleic acid had a profound effect on decreasing fibrosis at the histological level with decreased collagen production. Finally, we wanted to look at the effects of salvinoleic acid and CD36 antagonism on the fibroblasts that comprise the healing wounds. And we used fluorescence activated cell sorting to do this with labeling of known fibroblast markers. The main interesting finding from this was that salvinoleic acid treatment reduced the number of profibrotic reticular fibroblasts and increased the number of lipofibroblasts, which are less well known but are thought to be more anti-fibrotic. We also looked at the gene expression in the lipofibroblasts and the reticular fibroblasts and found that loss of JUN was associated with decreased expression of a number of genes highly involved in PPAR gamma signaling, which regulates fatty acid and lipid metabolism, and is therefore our hypothesis is that administration of salvinoleic acid decreased adipogenic genes involved in the PPAR gamma pathway, which links CD36 to adipocyte differentiation and suggests that CD36 may act via PPAR gamma signaling. This is outlining the conclusion to summarize our data altogether. CD36 is a mediator of JUN-dependent scarring via PPAR gamma signaling and acts at a cellular level to decrease lipofibroblasts and increase reticular fibroblasts. And salvinoleic acid can directly inhibit this pathway. Thank you to everybody who enabled this research to take place. It was a pleasure working with you all.
Terrific, thank you, Mimi. So the floor is now open for questions, and I'm going to start off by asking Ruth Tevlin. You had asked a question in the chat, and I'm going to invite you to unmute yourself if you don't mind, and repeat your question verbally, orally, and for Kellen Chen. Or I can just read your question, Ruth. So Kellen.
Oh, sorry.
Oh, okay. There you are. I didn't know if you were with us still or not.
Oh, yeah, sorry. I have two screens at the moment, so unfortunately I'm just doing something in the lab actually, but I was hoping that Kellen could elaborate further on his model where he had isolated the human fibroblasts and then placed them under strain in vitro. I know in the interest of time he glossed over that. I was hoping he could share a little bit more about that.
Yeah, Ruth, thank you for your question and your interest in the model. This is actually a model I developed in graduate school. And basically we take a cellular suspension and we mix it with soluble collagen, and that's kept cold. And basically if you heat it up to body temperature, that collagen polymerizes and you have a three-dimensional matrix where collagen is surrounding the cells. So the advantage is the cells are in a three-dimensional physiologic environment. It's all collagen like a scar. And then if we can attach certain if we can sort of fuse that with certain parts at the end, we can then pull on it and you can sort of get sort of stretch and mechanical activation. This is a softer collagen because that way it's able to polymerize. But we've been able to show that in this model with different amounts of stretch, you can basically modulate the amount of activation that the cells have.
Okay, great.
Thank you.
Kellen, it's Mike Longacre. That's also an advantage in cells that don't stick to plastic, right? So you're able to cells like adipocytes, for example.
Right, exactly. So that's something that could be used to encapsulate any type of cell, and you don't have to worry about them sort of sloughing off.
Thank you.
Great. And I know there was a question from Aaron Stone that Sandeep, you had answered, at least partially in the chat, but Aaron, do you mind asking your question again orally, and then maybe Sandeep can elaborate?
Can you hear me?
Yes.
Oh, so the question was, can you combine DFO with free fat grafting? Is there any potentiation between the two?
So yeah, thanks for your question. So we actually did a study, our lab previously back in 2018, and it actually checked on and evaluated the effects of DFO treatment on irradiated skin in mice and the subsequent fat volume retention. And we saw that DFO preconditioning does help fat graft retention, and there were some effects of also greater angiogenic potential and stuff of that nature. So yes, we've seen that in mice models, so we believe the same should be the case.
How much does it increase the take rate of the fat grafting?
The take rate. So if I remember correctly, the exact percentage, I think it was about 75% with DFO treated in comparison to just fat only, if I remember correctly. I should pull that up right now. I think it's a 50% fat volume retention over time with just DFO, sorry, without DFO, and with DFO, it's about 70%. So there was a significant increase, and this is over an 8-week
So it's
period.
increasing your take rate by 50%?
No, by 20%. So 7, yeah.
All right.
And this is just.
And do you have to get, was it proportional to the amount of fat, the DFO that you added?
So could you repeat the question? Sorry.
Was the amount of DFO that you added proportional to the amount of fat that you injected?
So in the mice models we used, the way we do it is we add 200 microliters of fat. And then for the injectable DFO, I believe, I don't exactly know since I didn't do the study, but I remember it was 1 mg per ml of concentration of the DFO that we inject prior to the fat graft. So I'm not exactly sure about the volume.
You're injecting them separately. They're not mixed together.
They're not mixed together. That's I don't think so.
Sandeep, it's Mike Longacre. I can help answer, or Dr. Wan. There are multiple studies published by Dr. Wan's laboratory, whether it's whole fat or subfractions of different populations within fat. And it's all highly significant, even in the face of radiation. But those studies have been published over the last 5 years.
The work that we've done with deferoxamine has been preconditioning the irradiated tissue before fat graft placement. And we've injected 1 milligram either directly into the subcutaneous tissue or we've delivered it transdermally also. And that subsequently has led to improved fat graft retention. Outside of our own lab, though, there have been other groups that have also looked at directly injecting deferoxamine into the fat graft after fat graft placement. And I believe their dosages are around 1 milligram also. And some of these reports have also suggested that there is improved fat graft retention with that, though there's also a lot of trauma associated with direct injection daily or every other day into the fat graft. And looking at histology, there are some evidence of some fibrosis also.
Okay, great. This is a terrific discussion, but in the interest of time, I'm gonna move on here, all right, and start our second part of our papers tonight. And is Dr. Rabbani on the line?
Yeah, I'm here.
Okay, great. All right, well, terrific. So pleased to introduce Pule Rabbani, who will be presenting her paper, Amniotic Fluid Stem Cells Ameliorate Wound Healing in a Diabetic Murine Model. And Dr. Rabbani is joining us from NYU.
Thank you, Henry. Good evening, everybody. My wonderful first authors are actually on to the next stages of their training. So I'm going to be presenting on their behalf. mean, with this audience, I don't think I need to introduce the public health issue of diabetes and its related complications. Something interesting looking through the stats was that apparently every hour, 10 Americans undergo a limb amputation due to diabetes and its complications. And we obviously have current management, but these wound healing complications persist and highlight the need for novel treatments to reverse this pathologic healing in this population with diabetes. These type of chronic wounds are stalled at the inflammation phase of the wound healing trajectory, indicated by that orange box in this figure on the left-hand side. And our current options are limited and mostly invasive. And like many other labs, we've been exploring viable options that could actually address the treatment of these ulcers. And for this talk, I'm going to focus on our work with amniotic fluid-derived multipotent stromal cells. I'm going to refer to them as AFMSCs moving forward. So what are these cells? They're a multipotent mesenchymal population. If you look at the figure on the right-hand side, they're more similar to mesenchymal stem cells, multipotent stromal cells. Take your, pick of name, as they're called these days. And they can be isolated from all the terms of gestation, but obviously a full term makes most sense, particularly at the time of a C-section. They adhere to plastic, also expand easily, and so far have been used in injury models of cartilage, muscle injuries. So we were approached by Dr. Bruce Young, who's an OBGYN at our institution, and he had been working with them for a while, but the cells hadn't been tested in any biological context. And that's where we came in. So we used the stented wound healing model everyone here is obviously very familiar with. And we applied a topical suspension of these AFMSCs, our vehicle, which is just the basal media being our sham control. And we photometrically observed the wound closure. And I'd like to draw your attention to the bottom row here that's treated with the AFMSCs. You see a drastic reduction, especially if you compare it to the vehicle control and the untreated diabetic wounds in the rows just above. We quantified this photometric data here on the left-hand side, and I think this makes it easier to appreciate the altered wound closure trajectory in the green line of the AFMSC treated wounds. And the bar graph on the right-hand side also shows this very significant change compared to the red bar or the yellow bar. And what's interesting for us is that this decrease in closure is bringing it closer and closer to a wild-type wound in the blue bar, indicating that some of the pathological aspects may have been reversed. So we went on to further confirm the photometric measurements of basically the scab, with measurements of the epithelial gap or the distance between the leading epidermal edges. One aspect that we were interested in is looking at whether these cells incite an adverse immunological response. And looking very early on after the topical administration, we find zero excess cellular influx of any sort. And despite the organismal mismatch, this is not surprising given the literature about mesenchymal populations like this, but definitely helpful for our project. We want to further explore how these AFMSCs might be orchestrating this rapid healing phenotype. And this is one of our more interesting findings, that on the right-hand side is the treated one, on the left is the vehicle. And on the right, we find much more expanded granulation tissue below the white dashed epidermis boundary there. And this has presence of a large number of F480 positive macrophages, but also arginase 1 staining, co-staining F480 positive macrophages, which are used to identify pro-repair or anti-inflammatory macrophages. So this was in the wound edge leading into the bed, and we found a similar presence in the wound periphery. And by that we mean the intact skin just past the proliferative parts of the epidermis, moving away from the center of the wound. And this led us to think of a probable cellular mechanistic event that might be happening to push the chronic inflammation down with the application of the AFMSCs, and then promoting proliferation and then remodeling. So I cut out a bunch of slides in the interest of time, so I'll move on to my summary. That these AFMSCs, they're very widely available worldwide, easy to isolate, so a terrific therapeutic cell population, and a topical and a local AFMSC dose can effectively drive wound closure in a mouse diabetic wound model and normalize the pathologic wound trajectory. And this therapy also seems to promote expansion of pro-repair macrophages within the granulation tissue and the surrounding skin. So before I end this, I have to thank these three bolded names up top. They executed the study, wrote it up, and the rest of my team, as well as our collaborators and our funding sources for this study. Thank you for your attention, and I'm happy to answer any questions on the chat or afterwards.
All right, well, thank you, Sheila. Our next paper is going to be given by Malini Chinta. Are you ready?
Yep. Okay, great. So go ahead and share your screen, and Malini will be talking on a presentation entitled Dermal Wounding Reveals Focal Adhesion Kinase Dependent Tissue Resident Fibroblast Progenitors. And Malini's speaking from Stanford. Go ahead. Malini,
hi, everyone. Oh, can you hear me okay?
Yes, now I can.
Great. Hi, everyone. My name is Malini Chinta, and I am a fourth-year medical student at Boston University. Today, I'm going to be speaking about some of the work I completed during my 2-year research fellowship at the Longacre Lab at Stanford University. Thank you all for the opportunity to present today. I know we've seen some of these images today earlier, but scarring in the skin can result in lifelong disfigurement and disability, and effective wound healing therapies have remained elusive. In this project, we hypothesize that there are tissue-resident precursor fibroblasts that are activated in the context of wound healing and fibrosis. We sought to determine the role of mechanotransduction on this phenomenon with the hope of informing the discovery of novel therapeutics. So this is a picture of the wound healing model that we use within our lab. Our wound model involves the use of a stented silicone ring around the mouse wound, which is used to mimic the tight-skinned human wound healing environment. In order to answer the questions about the origins and activation of wound healing fibroblasts, we used a rainbow mouse model. The rainbow model is a 4-color reporter construct depicted in the top right-hand image here, which in simplified terms allows us to precisely lineage trace fibroblasts. We created wounds on rainbow mice and then harvested these wounds at various time points throughout the wound healing process in order to assess the proliferation of these fibroblasts in vivo. So after harvesting our mouse wounds, we looked at them using a confocal microscope. The image on the left is a confocal image of a hole-mounted rainbow mouse wound at postoperative day 14. As you can see, there is clonal proliferation within the dermis, which is evidenced by the expanding fluorescent colors. We also saw that this clonal proliferation extends from the periphery of the wound towards the center, which is evidenced by the white arrows. So this suggested to us that the presence of tissue-resident fibroblast progenitor cells are activated in the context of wound healing. On the right-hand side here, we have a comparison of wounded skin versus unwounded skin. And as you can see in our wounded skin in the top panel here, there is increased clonal proliferation within the dermis, which is evidenced by the increased size and amount of clones when compared with the unwounded skin in the bottom panel. So to begin to genetically characterize these wound healing fibroblasts, we performed bulk RNA sequencing on our mouse wound specimens. On PCA analysis, which is shown on the left here, we saw that mouse wound fibroblasts cluster distinctly from unwounded fibroblasts. Furthermore, we saw that inner wound specimens clustered distinctly from outer wound specimens, and this can also be visualized on Pearson correlation. We then performed single-cell RNA sequencing on our rainbow fibroblasts, which we had isolated from our wound specimens in order to further genetically characterize these cells involved during wound healing. Clustering of this gene expression dataset revealed 4 distinct populations of fibroblasts, which is shown in the middle panel here. Of note, in cluster 0, we saw an upregulation of elements in the focal adhesion kinase pathway, or FAK pathway, which is a pathway that's heavily implicated in mechanotransduction during wound healing. So given these results, we applied an FAK inhibitor to our mouse wounds, and we used an FAK knockout mouse model to determine its effect on clonality within the wounds. So the top image here is a control wound. The middle image is a wound that has been treated with an FAK inhibitor, and the bottom image is a wound that was created in an FAK knockout mouse model. As you can see, the clonal proliferation in the bottom 2 images is significantly disrupted when compared to the control image at the top. So these results suggested to us that the activation and clonal proliferation of fibroblast progenitors was in fact FAK dependent. In conclusion, we found that dermal fibroblasts in wounds undergo clonal expansion in the wound microenvironment, suggesting the presence of a tissue-resident fibroblast progenitor type cell. We found that this clonal proliferation stems from the outer edge of the wound and is regulated by local mechanical forces in an FAK-dependent manner. Future directions for this project include a more detailed epigenetic characterization to elucidate the fibroblast heterogeneity in this context, as well as identify potential progenitor phenotypes, which could be targeted specifically to improve wound healing and decrease scarring. I would like to thank the following members of my lab for their help with this project, and a special thank you to my PI, Dr. Longacre, and thank you to everyone listening today. I would be happy to take any questions.
Thank you, Malini. That was terrific. We'll move on to our next paper, all right? And that is going to be Mimi, right? Okay, Mimi. So Mimi Borelli will be presenting our next paper, and it is entitled PRX1 marks ventral fibroblasts with increased fibrogenic potential. Take it away, Mimi.
Thank you for having me speak again. I have nothing to disclose. Skin fibrosis amounts to significant morbidity due to the prevalence of traumatic burns in burn and traumatic injuries. Fibroblasts are chiefly responsible for extracellular matrix deposition in the skin and are increasingly recognized to be a heterogeneous group comprised of multiple subpopulations with distinct functions and scarring propensity. While anatomic and embryonic origin are known to determine fibroblast heterogeneity, most of the work in mice to date has focused on the dorsal skin. In this work, we wanted to investigate whether embryonic lineage and dermal spatial location define fibroblast heterogeneity in the mouse ventral dermis, both in homeostasis and in acute and chronic fibrosis. For this, we used a PRX1 Cree R26 MTMG double transgenic mouse. This mouse has, basically every cell in this mouse expresses either a green fluorescent protein on its membrane or a red fluorescent protein. If the cell expresses PRX1, this switches on Cree, which switches on the green expression. If it doesn't express PRX1 and it's PRX1 negative or PNF, it is red. Interestingly, we found when we looked at the ventral dermis throughout embryonic development, an increasingly larger percentage of fibroblasts in the ventral dermis were green and not red. And there was a key transition point at before and after E16.5, which marks the time point in embryonic development when the mouse skin starts to heal with a scar. So this highlighted PPF, PRX1 positive fibroblasts, as potentially more fibrotic than the PNFs. And we wanted to investigate this further with a model of acute fibrosis. We used the stented excisional wound model described earlier, and looked at the composition of cells in healed wounds after they'd healed at 14 days, both by microscopy and fluorescence activated cell sorting, and found that the wounds were extremely green and comprised mostly of PPFs and very few PNFs. Next, we looked at whether these PPFs also were responsible for chronic fibrosis in the mouse ventral dermis. We used a model of radiation-induced fibrosis and irradiated the right hind limb of the PRX1 mice with a total of 30 gray, and waited 4 weeks for the chronic effects of radiation to develop. When we looked at the skin histologically, we found that the irradiated skin was much greener than the non-irradiated skin, and confirmed this by fluorescence activated cell sorting at a cellular level. We used a second model of chronic radiation, chronic fibrosis. We injected melanoma cells into the ventral dermis and looked at the fibroblasts in the stroma surrounding this cancer, and found again that at both a microscopy level and by a cellular level with FACS, that the majority of the stroma comprised PPFs and very few PNFs. An interesting question you have to ask is whether it's the microenvironment or the cells themselves that are responsible for this pro-fibrotic effect. And the way to do this is to do a reciprocal transplant experiment. If you focus on the right, this is showing how we took fibroblast PPFs from the ventral dermis, so the pro-fibrotic subpopulation, and injected them into the oral dermis of recipient black 6 mice. And we waited and harvested this tissue and looked at the association with collagen, and found a very high degree of collagen association as shown in that diagram on the right, bottom right. When we did the reciprocal and took Wnt1 expressing fibroblasts from the oral dermis of Wnt mice, which are classically known to be non-scarring and in a very scar privileged area, and we isolate the green Wnt expressing cells and inject them into the ventral dermis, we found we could isolate a patch of green, but it had very little association with collagen and was not very fibrotic. Finally, we performed a diphtheria toxin ablation experiment. We crossed our PRX1 mice with mice that express a diphtheria toxin receptor to make a mouse which expresses the green fluorescent protein when it expresses PRX, and also expresses the diphtheria toxin receptor, meaning that we can selectively ablate the PRX1 positive cells. When we do this, if you look in the right, we find that the skin comprised red and not green, the scarred skin, and it was also less fibrotic. Interestingly, when we looked at the tensile strength of skin in the diphtheria toxin treated, which is the red line and the red bar, we found that that was stronger and more similar to unwounded skin shown in the green line and the green bar, compared to mice that had just received the control PBS treatment. And when we did this in our tumor model, we found that there was less PPS and less collagen, which collagen's in red in this diagram. So in conclusion, these data suggest that PPFs are responsible for both acute and chronic fibrosis in the mouse ventral dermis. And thank you to my wonderful research group and my wonderful mentors for making this research happen.
Okay, terrific. Thank you, Mimi. All right, our final talk for this session is entitled Epidermal NRF2 Orchestrates Tissue Regeneration Through Regulation of CCL2. And it'll be presented by Dr. Alvaro Villarreal Ponce.
Yes, thank you so much.
Okay.
Let me just share this. Share. Start. Could you see?
It's not in slideshow, but we do see your presentation.
Oh, really? Let me see if I could adjust that.
I think.
Is that better?
No, but I think this is fine. We can see slides. So.
Okay, perfect. Then I'll just proceed like this.
Yeah.
Well. Okay, well, thank you very much to PSRC for allowing me to share this work. My name is Alvaro Villarreal Ponce, and I am a postdoctoral research fellow at NYU Langone Health in the laboratory of Dr. Daniel Cherardini. So chronic diabetic wounds impose a major socioeconomic burden that costs over $40 billion annually and negatively impacts an estimated 30 million individuals in the US. There is an absence of effective treatments for this debilitating condition, in part from our reduced understanding of the signaling networks that are on set during wound repair, as well as how dysregulation of these pathways contributes to impaired diabetic healing.
Alvaro, are you advancing your slides?
Yeah.
Okay, that we're not saying.
Is there a delay?
Must be a very long delay if because it's still on your title slide.
It might be the screen that you're sharing. You might have. Do you have two screens up?
Let's see. No, I have the one that's covering my entire screen is the PowerPoint slide.
So, but that's not the one we're seeing. So when you click share screen, it can give you a couple screen options.
Oh, I see.
And we're seeing your desktop.
Oh, really? Okay. Let me cancel share. How's this?
That looks better. Okay, yeah, you can.
Okay. Okay, perfect. Sorry. Okay, so this is where I was at. And given the broad accountability of oxidative stress in many pathological processes associated with hyperglycemia, we focus on elucidating the function of NRF2, master regulator of transcriptional master regulator of oxidative stress in tissue repair. We have previously demonstrated that NRF2 dysfunction in diabetic wounds results from impaired activity of its native repressor, and showed that focused activation of NRF2 improves the regenerative potential of diabetic wounds. We turn our attention to uncovering the spatial temporal mechanisms that are regulated by this transcription factor. First, using the DB diabetic mouse model, we revealed using immunohistochemistry and functional analyses that the epidermis is a prominent compartment of the diabetic wound environment that fails to activate NRF2. Thus, to analyze the NRF2 function in this compartment, we generated conditional epidermal-specific NRF2 knockout mice, which we used to delete NRF2 in the epidermis of adult mice and analyze wound healing dynamics. Performing 10 millimeter stented excisional wounds on the dorsum, we found a delay in homozygous epidermal NRF2 mutants when compared to littermate controls that closely approximated the delay observed in diabetic mice. This delay associated with epidermal NRF2 knockout mice was depicted through analyses that showed reduced wound closure rate, time to closure, and reduced wound burden, which is representative of the regenerative potential of the wound. To gain mechanistic insight into this delay, we sorted wound-associated keratinocytes at 5 days post-wounding and performed bulk RNA-seq. Gene ontology of differentially expressed genes showed not only reduced expression of genes involved in oxidoreductase activity, but also keratinocyte-mediated paracrine signaling pathways involved in inflammation, immune cell guidance, among others. Interestingly, chemokine CCL2 was a downregulated gene that was recognized in many wound-related categories. Because of the known role of macrophage trafficking through the CCL2-CCR2 signaling axis, we quantified macrophage abundance in wounds in epidermal NRF2 knockout and control wounds over time. While no difference was found in intact skin, there was a dramatic 80% reduction in the number and density of macrophages in knockout wounds by day 5, which persisted to later stages. Having identified CCL2 as a putative NRF2 target through our RNA-seq, we confirmed its regulation by activating NRF2 with triterpene molecule CCDOIM, which elevated CCL2, mRNA, and protein expression. Next, using chromatin immunoprecipitation on keratinocytes extracted from intact and wounded skin, we showed the dynamic binding of NRF2 to specific binding regions of the CCL2 promoter in the wound setting, but not in intact skin. Next, to determine whether CCL2 is responsible for this wound repair phenotype observed in the epidermal NRF2 knockout mice, we applied recombinant CCL2 onto wounds and assessed how the new wound healing dynamics. As shown in the aqua-colored arrows, CCL2 treatment to epidermal NRF2 knockout wounds restored the wound closure rate, time to closure, and wound burden near physiologic levels, suggesting the importance of CCL2 regulation by NRF2. While not shown here due to the time limitation, we also showed the reduced CCL2 expression in the wound-associated keratinocytes of diabetic wounds, and we were able to restore the diabetic healing through exogenous application of CCL2 as well. So in summary, we provide evidence showing the importance of epidermal NRF2 in repair and uncover a significance of the NRF2 CCL2 signaling axis in coordinating the cellular crosstalk between keratinocytes and macrophages. Thank you very much to NYU and Dr. Ciardini's laboratory, and thank you very much for the time.
All right, well, thank you, Alvaro. All right, that concludes the session, but we are open for discussion, and I will actually ask, there's a question I got privately. I'm going to ask questioner Nicole Gerlone. You have a question for Dr. Rabbani, is that correct?
Hi, Dr. Rabbani. Thank you very much for your discussion. I was wondering if your team observed a dose-dependent relationship between the amniotic fluid stem cells and either the macrophages or wound closure, but you might have touched upon this a little bit in the chat already.
Yeah, I think Kellen caught on the same stuff. We tried a couple different doses, and we have some experience with other mesenchymal cell types also. And we find that we went everywhere from like 1 million to 10 million and beyond. And we end up finding that beyond 500,000 or 1 million or so, the effect's exactly the same in terms of the wound closure. And I think part of it has to do with access of the cells to the exogenous ones being applied. But the other is we're hitting physiological limits of the tissue as well. Because if we fast forward it a little too much, we're not gonna get any of that collagen granulation tissue wall built. So I think that's what we're up against. And we're okay with that. We have to give the skin time to like put its bricks in, right? To build that.
Thank you.
So we are a fair bit beyond our scheduled time. So unless someone has a burning question to ask, I'm gonna close the session, thank all the speakers, congratulate them on terrific papers, and thank everyone for joining us. And I'm going to hand this back to Derek.
Yeah, thank you, Henry, and thank you. These have all been phenomenal talks. And again, I'd like to thank all the participants tonight, as well as Dr. Shah for moderating. Once again, I also want to thank 3M for their generous support of tonight's program. And I'm really looking forward to the upcoming talks by both Dr. Gertner and Dr. Wong this coming Thursday, so please log on to that also. Finally, as I previously mentioned, we will now transition to an informal mentoring session. So for any remaining active members of the Research Council and all their attendees, please stay on if you're free. So thank you again for the wonderful evening, and I look forward to seeing you all on Thursday.